Assalamualaikum and good morning to everybody. My name is Nur Husnina binti Azad, 194748, and my supervisor is Dr. Annie Christianus. Today, I'll be presenting about my final year project on effect of feeding rate and thyroxine immersion on the molting of horseshoe crab Tachypleus gigas larvae. So this is my table of content. Okay, around the world, there are 4 living horseshoe crab that can be found. And two out of 4, the coastal horseshoe crab Tachypleus gigas and the mangrove horseshoe crab Carcinoscorpius rotundicauda can be found in Malaysia. Well, according to IUCN, animals such as sponge, mud crab, mussel, and snails use the hard shell of the horseshoe crab as microhabitat. Therefore, it shows that horseshoe crab is a vital link for the coastal biodiversity. All right. According to Button and Shin 2020, um, the 2 Asian horseshoe crab will soon be listed on the IUCN Red List. So this is due to the decreasing number of horseshoe crab around the world due to different factors such as water pollution, the loss of their habitat for living and also spawning, and not to forget the overharvesting of the adult horseshoe crab. All right, the my problem statement for this project is this: there first is scarcity of report on larvae culture, especially on feeding rate effect on the growth of Tachypleus gigas larvae. And next is there is actually no report on effects of thyroxine hormone on molting of Tachypleus gigas larvae. All right. My objective for my experiment is to determine the average of Artemia nuclei consumed by horseshoe crab larvae per feeding, and to observe the determine the effect of thyroxine T4 hormone immersion on the molting of horseshoe crab larvae. Let's move on to literature review. Um, Tachypleus gigas, Tachypleus gigas come from the phylum Arthropoda, which is known with their characteristic, which is they have a very hard exoskeleton, and they come from the family Limulidae and genus Tachypleus, based on Muller, 1785. And Tachypleus gigas can be found in East and West Peninsula Malaysia, and they prefer to live in sandy and muddy condition. And Tikkikas is not tolerant to a low salinity because they need sandy beaches with high salinity in range of 31.8 ppt. Okay, for the behavior of the Tachypleus gigas, adult horseshoe crab migrate from the offshore continental shelf to spawn on intertidal sandy. This is a case for Tachypleus gigas, and usually they always comes to the beach to spawn at every full moon and new moon phase. Okay, for the feeding behavior, um, Tachypleus gigas is actually a selective feeder, and it has been analyzed from their gut content that they actually prefer mollusk and decayed organic matter. Um, but there's shows that from Mishra 2009 that trilobites of Tachypleus gigas in a controlled environment, in lab environment, they can be cultured in a controlled diet of brine shrimp, Artemia, and also diatom of Cetiosaurus gracilis. And the average total length of freshly hatched trilobite larvae, the one that does not have any tail yet, would be 8 millimeter in length. And the average grow rate from first to second post hatch stage will be around 63%. So, and the thyroxine hormones plays an important role in the process of metabolism, absorption of the yolk sac, and fish growth. This is why I actually use tyrosine hormone in my project. So move on to methodology. So the horseshoe crabs eggs were actually being collected from the wild at Sandy Beach at Kampung Batu Lapan, Tanjung Sepat, Banting, Selangor. So the sample and feed preparation for my experiment, after the eggs were collected, we actually get 4 batch of eggs, and all these 4 batch were washed and filtered with seawater and fresh water respectively to remove all the sand and debris that attach to the eggs to make sure it is clean. And most eggs that is used in this experiment hatch on 50th to 20th September of 2020. So the location for the my experiment has been conducted at the Institute of Bioscience IBS Hatchery at University Putra Malaysia. So preparation of treatments for my both of my experiment is for experiment 1, 3 petri discs used for each treatment, which is treatment 1, treatment 2, treatment 1, treatment 2, treatment 3. And for experiment 3, there would be 3 treatment for thyroxine concentration, which is 0, 0.5, and 1 ppm. And another factor is number of immersion, which is 1 time, 2 time, and 3rd time. And each factor have 3 replicates. And water exchange is done every 2 days. So for the feed preparation, for both of for all my experiment, I've been using Artemia salina. So to prepare my Artemia, I'm using a conical bottom as hatching tank, which was clean and filled with filtered seed water. Salinity required would be along 15 to 30 ppt, and temperature would be around 25 to 30 degrees Celsius. And every time half a teaspoon of eggs were hatched within 24 to 36 hours. So this is my experimental design. As you can see, each treatment have 3 replicates, and each petri dish have contain 5 horseshoe crab larvae. Artemia hatch was carried out once in 2 days using Artemia egg with a weight average of 0.123 gram, half a teaspoon, in 500 milliliter of sterile and filtered seawater. So there's 3 treatment: treatment 1, treatment 2, treatment 3. So for treatment 1, every replicate will be fed around 500 nuclei. For treatment 2 will be fed 750, and treatment 3 will be fed around 1000 Artemia nuclei. Okay, for next experiment, which is dosage and number of immersion. First, for dosage of thyroxine hormone, there are 3 treatment, which is control 0, 0.5 ppm, and 1.0 ppm. The preparation of thyroxine were conducted at the lab in Faculty of Agriculture in aquaculture department. So the thyroxine was weighed and dissolved in 1 liter of sterile and filtered water. So the 2 concentration of T4 were prepared, which is 0.5 and 1.0 ppm. And pH of the solution were adjusted between 8.0 and 8.5 with sodium hydroxide using pH 700 meter. So this is my experimental design for the second experiment. So first, my data was collected from 9th of November till 50th December on 2020. So as you can see, the blue color indicate the petri dish of 1 ppm, the yellow color is 0.5 ppm, and the green is control. So each 0.5 and 0.0 have a different number of immersion, which is oh, 1 time, 2 time, and 3 time. So the first time immersion of T4 immersion is on 9th of November. Then the second time of immersion will be on 60th, and the third time will be on the 23rd of November. So my data analysis, for my data analysis, um, experiment one has one factor, so I'm using one-way ANOVA, and experiment two will be using two-way ANOVA, due to they have actually two different factors that affecting the results. So both of the experiment I'm using IBM SPSS statistics version 25 with two key new multiple range tests for my post-hoc test. Okay, this is my result for feeding experiment. As you can see in the table, there are feeding rate, which is number of the Artemia nuclei per feeding, and also percentage of lava molted in percentage. All data you see here is actually has been transformed to arcsign data, and it is in form of mean plus minus with standard deviation. So after running the test, the one-way ANOVA, it is shown that the p-value was higher than the 0.05, which is the p-value is 0.943. So therefore, there is no significant difference on the percentage of molted horseshoe crab larvae that observed between all three of the feeding rate. Okay, for my effect of dosage of number of immersion of thyroxine experiment, this my data in this table also, I've been transformed the data to arcsign, and as you can see, they have T for concentration, and number of the immersion, and also the percentage of lava molted. So this data is also in terms of mean plus minus with standard deviation, and as you can see, for all three, p-value is all higher than 0.05. Therefore, there is no significant difference on the percentage of molted horseshoe crab larvae observed between all treatment of different concentration and number of concentrations. So for the discussion for my result is based on experiment 1 result, the different feeding rate used Artemia nuclei showed no significant difference on the percentage of molted Tachypleus gigas larvae due to no effects on its growth. All three feedings, 50, 500, 750, and 1000, showed similar growth, indicating even though it was more than it shows that the feeding is actually over its limit feeding capacity, which is also mean it's not optimal for Tachypleus gigas larvae. Therefore, for future research, it's possible to look into reducing the number of Artemia per feeding to determine the optimal rate for this horseshoe crab larvae. For my experiment 2, there's two factors. But first, we go to the concentration of T4. So it shows that the different T4 concentration showed no effects on its growth. Thus, of course, also there is no significant effects on the percentage of molted Tachypleus gigas larvae. Well, as I mentioned earlier, Tachypleus gigas come from the phylum Arthropoda. So the species has this hard exoskeleton, which is the physical, which maybe could be the this physical feature could have the limit the absorption of T4 during immersion, even with different dosage of T4. Thus, it shows no significant effect, even when given at the concentration, may contribute to enhancement or inhibition of growth growth, such as other species, which is pink zebrafish larvae, Branchidanio. Well, therefore, this study was conducted to determine whether T4 can speed up the growth of horseshoe crab or not. As it is, horseshoe crab actually takes about 9 years to only reach its puberty. Therefore, its growth can be significant. If the growth can be significantly enhanced, um, then the culture to culture the species may be viable. Okay, for the second factor, which is the number of different number of T4 immersion, also result in no significant difference to molting of Tachypleus gigas larvae. Similar to the dosage experiment, the hard shell of horseshoe crab hinders the absorption of hormone efficiently into the body as compared to other fish species. So further investigation need to be carried out if immersion duration can significantly contribute to enhance of growth of this horseshoe crab. Longer duration of immersion may help this hormone to penetrate the shells better. So at 1 ppm T4 can actually inhibit the growth or worse increase the mortality of fish fry, according to DGS's Toledo and Simpas on 1998. However, this concentration actually showed no adverse effect on the horseshoe crab larvae. Plus, as the hormones is costly, using T4 at high concentration is not economical. Then, for further study, I hope it is possible for the further study. It is possible to experiment more on length time of immersion of the T4 hormone on the Tachypleus gigas larvae. So my conclusion is, for my experiment, for my project, feeding rate at 500 to 1000 atm apply per feeding, and T4 immersion at zero point 0.5 to 1 ppm does not affect the growth of Tachypleus gigas larvae. This is my reference. So thank you.